On September 19, 2003, the Albert Lasker Basic Medical Research Award was bestowed on Robert G. Roeder of Rockefeller University for his pioneering studies on eukaryotic RNA polymerases and the general transcriptional machinery which opened gene expression in animal cells to biochemical analysis. Robert Roeder is interviewed by Lasker laureate James Darnell of Rockefeller University.
I'm Jim Darnell, professor at Rockefeller University, and I have the distinct pleasure and privilege this morning of having a chat with my colleague, Bob Roeder, who is the winner of this year's Lasker Award for Scientific Achievement. A special friendship with my colleague Roeder began many years ago, before he moved to Rockefeller, 20-odd years ago. And we're going to have a chance to hear him tell us how he began his scientific career, and we will introduce you through his eyes to some of the events that have occurred that he feels is significant during his more than 30 years of studying how cells make RNA. Bob, I know that we've shared a lot. I've been diddling with RNA 10 years longer than you have, but it was a very important time in the late 60s when your first important publication came out. And we certainly want to get to that. But we share something else, which is we both came from very small towns, as we've talked about many times. And I think people who may see this might be interested to know how a young fellow born in Boonville, Indiana, got started in major medical research. What happened in Boonville that interested you in science?
Well, actually, nothing happened in Boonville that interested me in science, because I moved at the beginning of high school from Boonville. But I will tell you a little bit about Boonville. It was a farming community founded by not the famous Boone, but by his brother Ratcliffe, and was largely a farming coal mining community. And I had the standard education. The hardest education was on the farm, because my father was a farmer, had a small grain farm, and I learned how to work hard on the farm. But I did enjoy school, and my freshman year in high school, we moved north to a town called Jasper, Indiana, or near Jasper, and that was where I completed high school. And that was where I began to take an interest in some of the scientific, in some aspects of science. I was interested in mathematics, I was interested in chemistry. Biology wasn't then of much interest because it was so descriptive and memorizing things. I much preferred in chemistry to do things with my hands and mix things together. But that basically was my first exposure to science, and in terms of a future career in science, I hadn't thought much about it until the end of high school, when I was struggling to think about something to do. My father was very determined that I should stay on the farm, so much so that he had nothing to do with my college education. In fact, did his best to keep me on the farm. But I escaped to Wabash College, which is now a men's school, and had fabulous science departments in biology, chemistry in particular, and in math. I started out as a math major, but it became clear I wasn't a genius in math, because many of the kids there had, from private schools particularly, had advanced training in mathematics, and I really developed an interest in biology and chemistry just because of the excellent teaching of people there.
Mm-hmm. We don't want to make a sociological tract out of this, but I'll ask you one question. What do you think would happen to a youngster these days, born in Boonville, moved to Jasper? I've often wondered this from my own background also. What's happening in mid-America, small-town America, in terms of education? Do you know?
Well, in my own high school, I know for a fact that it's much more modernized in terms of teaching biology and molecular biology. In fact, one of the people I played football with in high school, and I can tell you I was better known for playing football in high school than I was as a scholar, even though I was at the head of my class. So my friend who was a football player with me, in fact, ended up getting a degree in biology and becoming a biochemistry teacher in high school, and was very interested in having me come back and talk on occasion to the students. So I only wish they had been so up to date, I think, when I was there.
Good. So we're not going to lose the next generation of Raiders. So.
I hope not.
All right. Let's move on beyond your successful time at Wabash College to the time when you started to get your PhD. I know that you went to Illinois to enroll in biochemistry to begin with, and you've told me that there were famous professors that you had a chance to meet there, but let's skip that for a second, because it was after your mentor, Bill Roeder, moved to the University of Washington that things began which are relevant to what we'll talk about the rest of our time. Tell me what it was like in Roeder's laboratory, and in particular, reflect on what you and Roeder and others knew and thought about how animal cells make RNA at that time.
Well, Bill's lab was in a transition at that time. He was better known as a classical enzymologist, particularly his work on aldolase. And in the last year at Illinois, when I joined his lab, he had just come back from a sabbatical at Cliff Grovestein's laboratory at Stanford, and who worked on the pancreas system as a developmental model. And Bill saw this as an area of major interest for him in the future, decided in terms of looking at developmental programs to start by measuring, developing sensitive assays for all of the pancreatic enzymes, the secreted proteins and such, and had visions really of studying RNA at the time, although nothing was going on in the laboratory. The work was split between aldolase biochemistry and pancreas differentiation. Bill had given me an opportunity to, thinking that down the track he would study transcription problems in the pancreas, had given me an opportunity to join the lab to hopefully find a problem that would ultimately end up at that junction. The lab was a very exciting laboratory. Bill was one of the more dynamic and stimulating people I've ever encountered in science. Everyone had sort of freedoms to do as they wished, although there were regular group meetings and periodic meetings with him as well. But in the animal cell world, this was at a time when your own work was really making seminal observations about hnRNA. The messenger concept was well established in bacteria, but obviously no messengers existed in animal cells, only the major classes of RNA, including hnRNA. And we had meetings with others at Seattle who were interested in assays for RNA. In particular, Ben Hall, who had developed some of the hybridization assays with Spiegelman, was using that technology mainly in phage systems. Brian McCarthy had come from terrestrial magnetism to Seattle, to the University of Washington, to study, again, to begin to look at populations of RNA by DNA-RNA hybridization. Was mainly doing work in sea urchins with the Whiteleys, but it was clear to, I remember a particular meeting in which I actually presented work. This was a meeting between the Roeder, Hall, McCarthy labs, those interested in the sort of new field of gene expression in animal cells. And I presented the work of Don Brown on his studies of gene structure and measuring populations of RNA in animal cells, in this case during developmental transitions. And I recall one of the aforementioned individuals making the remark at the end, is that all that's known? It literally was the Dark Ages. Mm-hmm. And we were all anticipating, of course, that the messenger hypothesis would be validated, and waiting for people like you to solve some of those problems. But it was still, the field was limited by not having genetics in animal cells or by the complexity of the genome, because the hybridization experiments were really not measuring what people wanted them to measure.
Of course, that's right. As the scientists who may look at this will know, what you decided to do and successfully did was to study the enzymatic synthesis of RNA. Can you remember a time when that was decisively decided as what you were going to attack?
I can, because the initial studies in the Roeder laboratory, there were a number of diverse and failed studies that I won't mention, but some of the ones closer to transcription had involved using isolated nuclei from animal tissues, such as rat liver during hormonal responses, to measure RNA synthesis, and two things: to measure the overall level of synthesis, or to measure, hopefully with the hybridization, new populations that were induced. I remember in that case, the only thing I could conclude that was induced was ribosomal RNA, and that is true. That was a major RNA population which was induced in this response. But in terms of thinking about the enzymology, the studies in isolated nuclei were useful, but not giving me any insights. I have to admit, being biased by being in an enzymology laboratory where people talked a lot about enzymes and purified them, and there had been some speculation from studies by Widnell and Tata, who had looked at RNA synthesis in isolated nuclei under different divalent metal ion and salt concentrations, and under one set of conditions, the RNA had a ribosomal-like base composition, and in the other case, it had a DNA-like or an HNRNA-like base composition. So the speculation was there might be different enzymes. So this was intriguing to me, but the simple fact was I thought that the things I was doing were really not leading to an understanding of a transcription mechanism or even its regulation, and I simply began to think that looking at the enzyme or the enzymes was the way to go. And at that time, purified genes were available from the Brown and Bernstein laboratories, only the ribosomal RNA genes, and I certainly thought down the track there would be an opportunity to reconstruct a specific transcription event in a test tube.
At that point, were you, or was the group reflecting on any of the successes, by that time not complete, but beginning successes in studying DNA replication? In other words, that's a complicated multi-enzyme, multi-protein task also. Was that important in your thinking?
I think there wasn't, in the Roeder laboratory, much discussion about that. But in those days, I, much more so than now, was conscientious about covering the literature. It was easier in those days, but had other things to do. But I did follow the enzymology, and particularly the work from the Kornberg laboratory, where it was clear you could study enzymatic reactions relating to transactions on DNA. Mm-hmm. And again, the enzymology was a key there. The difference, of course, was that enzymology was carried out in bacterial systems where the enzymes were much more abundant than in animal cells.
Right, right. So finally came the time when you got solubilized enzymes. And tell us about the first year or two after those discoveries.
Well, the discovery of a solubilized enzyme came, that is the obtaining the solubilized enzyme, that happened in really January probably of 1969. I had spent a little time trying the classical approaches that the bacterial people used, which were to grind up the cells in a low-salt buffer, sonicate them or run them through a French press, and spin out the debris. And there was a soluble enzyme, and the bacterial enzyme was 1 part in 50. It had only to be purified 50 or 100-fold, perhaps. That approach did not work in animal cells, and indeed there were at least a half a dozen reports in the literature on studies of a soluble mammalian enzyme, but as it turns out, this was probably a few % of the polymerase, because most of it is bound to DNA and engaged in transcription complexes, and that, of course, is the basis of the nuclear run-on assay that you and I appreciate as the major means to measure an absolute rate of transcription. So after fussing around with techniques and having so little soluble enzyme, I decided there had to be another method. And in reading the literature about nuclei, it was clear that it was bound to chromatin, or to DNA in the form of chromatin. And chromatin was sort of a dirty word in those days. There was a lot of messy work on it, but it's clearly something that had to be dealt with. And I realized that I had to get rid of the histones and the DNA and get the enzyme solubilized. And I read a paper of Alfrey and Mirsky from the Rockefeller University that was dated 1942, in which they'd studied nucleohistones and realized and shown that high salt would dissociate the histones, and if they did a rapid dialysis or dilution, the DNA and the histone would rapidly precipitate from solution. Of course, they weren't studying RNA polymerases, but that particular insight was a key to getting the enzyme solubilized, because it ended up being the approach I used to simply take nuclei, dissolve them in high salt, sonicate them, which disrupted the DNA and reduced the viscosity, so you had a solution you could work with. But everything was soluble at 0.4 molar ammonium sulfate, and then the trick was to dilute it rapidly to 0.1 molar ammonium sulfate. DNA and histones rapidly aggregated. The polymerase was in solution.
And so the discovery that there were several polymerase activities followed that solubilization. And there's a famous chromatogram of 3 different polymerases, which I've taken advantage of, as you know, put it in a textbook. And was that a eureka moment for you when you saw the 3 polymerases coming off the chromatogram?
That definitely was a eureka moment. Not the last, but certainly the first, and perhaps the most exciting, because it really launched, in my view, launched the whole field. Again, a few people had been studying the animal cell polymerases, but really not enough enzymatic activity to work with. And ironically, even though at the time there were many studies ongoing of bacterial RNA polymerase, as you know, the first report of a bona fide RNA polymerase activity, period, was Sam Weiss's description in 1959, a report in the Journal of the American Chemical Society of an aggregate enzyme. It wasn't a soluble enzyme, but it was an aggregate enzyme. So the chromatogram you referred to was really a turning point for me, and happily it happened very quickly after I had a soluble enzyme.
Now,
and I have to admit that the trick to seeing 3 polymerases on the particular ion exchange resin I used was a result of my knowing that Cefidex A25 had a very high resolving power, because my good friend Ed Penhote had used it to resolve 5 aldolase isozymes, or hybrids of 2 different aldolases, and had recommended it.
So just to remind anyone who may eventually be looking on here, we're still, I won't say stuck, but we have spent a considerable time reflecting on the bench activity when Bob Roeder was a graduate student. This all occurred, and the discovery of the enzyme occurred. 3 enzymes occurred when he was a graduate student. Let's move on, skipping a bit, because there's something that I have always thought was of great importance in the program that has led you to where you are now. And that is the ability not just to observe the incorporation of ribonucleotides into RNA, but to make certain that the RNA that was being formed in the reaction started at the right place. Initiation demands that you start at the right place. And that happened first with polymerase 3, but then it also eventually happened with polymerase 2. And I think it would be worthwhile if you would just recount for us the correct start site thesis and why this is so important for subsequent work.
Right, but let me interject a moment to cover 2 years of my life as a postdoc. Having 3 enzymes was exciting, and the next step was to try to do what you suggested, to get the enzyme to recognize if there existed a specific start site, and the best candidate of the day were the ribosomal genes because they had been purified and fairly well characterized, especially by Don Brown, where I went for a postdoc. And I suspected strongly that one of the enzymes, RNA polymerase 1, was the correct polymerase because it was localized in the nucleolus, the site of ribosomal RNA synthesis. So I packed my bags, went to Don Brown's lab, purified the Xenopus enzymes, not to homogeneity, but adequately, I thought, for transcription studies. And one of the great disappointments and surprises and disappointments was finding that my purified polymerase 1 did not work on purified ribosomal RNA genes, leading me to suspect either there wasn't a really specific initiation site, although that really didn't seem probable. It seemed like something might be missing, or there was still a lot of attention to chromatin in those days, that maybe one needed a chromatin template to restrict the activity of the enzyme partially. But so that experiment did not work, but at Washington University, once we had established through studies with the mushroom toxin alpha amantadine that the 3 different enzymes really did have 3 different functions, and that's another sort of topic. But the point was we could then focus on POL1 on ribosomal genes, or POL2 on genes transcribing messages, or POL3 on on genes encoding small structural RNAs. And there really was a question in those days about whether there was a specific initiation site. Of course, nothing was known early on about genes for proteins, that is, genes encoding proteins through messages, but we did look at those initially, but we also began to study the 5S RNA genes because Don Brown had isolated them even before the cloning days as a pure population. And this was a short RNA, the gene being only 100 or so nucleotides long, but the key feature that was known about the 5S RNA was that it had a 5' triphosphate on the end of the RNA, which meant that was the initiating nucleotide, and it wasn't likely to be processed. And of course, processing was what we worried about, that the stable RNA species were derived from processing events. We would never know where to look. So it is true, as you hinted, that we got initiation to work first on these small genes, but it wasn't a pure enzyme on a pure DNA. It was a pure enzyme on a chromatin preparation from oocytes which had 30,000 active 5S RNA genes. And the polymerase initiated and terminated correctly, but it depended on chromosomal brown proteins, which we later characterized.
That did set the stage, however, for was the formal proof that you could, in a broken cell preparation, correctly start and stop an RNA.
No, it was key because it gave us confidence. I mean, the work proceeded there to showing that actually cloned genes could be transcribed in extracts. And after our initial description of success with a purified enzyme, 3 laboratories basically showed that a clone gene could be accurately transcribed in a mixture of, in an extract. Don Brown's lab, Guangxue Wu's lab for the adenovirus VA RNA, and our own lab. In our special case, we were able to show that the polymerase would transcribe the gene or initiate at the promoter, but only with some soluble proteins, which we had purified from the extract. But that definitely gave us confidence that the more interesting, at least to you, the more interesting class of genes might show the same success, given an ability to sort of know what to look for. And that's where your own work was so pivotal for us.
Yeah. Why don't you just briefly go over the starting that Tony Weil in your laboratory made on polymerase II genes, the genes that encode proteins.
So here, again, the key question was, what is a start site? We weren't so concerned about termination sites, but is there a specific start site that we could hope to use to reconstruct initiation? And adenovirus was sort of the genome of the day because it was a viral genome that could be chopped up into pieces with restriction enzymes and parts of them cloned. And the real question was, is there an initiation site? And of course, your own studies using a variety of techniques, nuclear run-on assays, UV mapping, and so forth, pinpointed on the adenovirus genome several initiation sites, but the one that's near and dear to us, the adenovirus major late promoter that you map to position 30 or so on the, 16?
Yeah.
16.
18.7.
18.7, sorry. Gave us a key, gave us a clear picture of where to look for a specific initiation event. And I have to tell you, one of the other eureka moments, of course, was our seeing that initiation event established, and that was the work of Tony Weil, who used the strategy of purified DNA fragments, purified RNA polymerase II, and extracts from the cell. And I remember the first time I knew the strategy that was ongoing, and it was a simple one. The sort of low resolution analysis specific initiation was simply to look for an RNA product that had the same size as the predicted size from the initiation site to the termination site, which in this case was the end of the DNA of a restriction fragment, so a runoff product. And I remember coming back from one of many seminar trips to an excited Tony Weil, who had to show me the runoff transcript. In fact, that event convinced me I should travel more because it was really an exciting event. This is what everybody in the field was waiting for. We were all interested in the small RNA genes as models, but of course the diversity of the genes encoding proteins made them of more intrinsic interest.
You've explained it clearly, but in the possibility that there are students who may look at this, let me reiterate what Dr. Roeder said. At this point, total extracts of nuclei plus purified polymerase plus pure DNA allowed correct starts, and those you will recognize are the ingredients for all the purification and identification of factors that has followed. Now, we've taken a good bit, perhaps close to most of our time, to get to this stage. The events after this accelerated. More labs began to work on it. And now we have reached the point where a great many, perhaps we'll never know all, but a great many of the proteins that participate in starting RNA synthesis have been discovered, a large fraction of them in your laboratory. We certainly don't have time to go through step by step all of these. But I wonder if you would like to try to characterize this era in which so many different proteins have been shown to play a role in getting this great machine to make RNA, which turns out, messenger RNA in particular, which turns out to be a key enzymatic process in controlling all of life's diverse events. go from the time when you could start correctly?
Right. There are several levels to that question, but the first one, of course, was what to do when we have specific initiation working with the purified polymerase. The first question, of course, is what are the other factors? And it was clear from those initial experiments, by the way, that whatever those factors were that allowed the polymerase to work correctly were general initiation factors, because remember, the adenovirus major late promoter is only expressed late in adenovirus infection after other viral proteins have carried out certain functions. And we were seeing initiation in extracts from uninfected cells. So we were pretty confident that these were simply general initiation factors that recognizing what we've come to call the core promoter. So there were two routes to go. One was to use that system to map promoter elements, which most labs did because it was an easy assay for those good with recombinant DNA. We chose the more challenging and harder route, and to me the more important one at the time for us, of going to the cold room, further fractionating the extracts, defining the factors. So that led to the definition of the general initiation factors. We started the process. Many people followed suit, a lot of former postdocs as well as other laboratories, to elaborate and elucidate the nature of the general initiation factors, which of course are a large number. Our own studies had also identified the factor that recognizes the core promoter, the TF2D that recognizes the Tata box in the case of polymerase II. So that was really a start, and said, well, there's a second level of control.
So by this time you had how many polypeptides on your hands? 50 at that
point?
Well,
the we includes the rest of the field for the,
I understand
for the simple reason that the pre-initiation complex for a gene encoding a protein is 44 polypeptides.
44.
If you include the polymerase.
Right.
And these are, and that we knew from the structure of the enzymes. We had in 1974 witnessed the first complete comparative analysis of polymerases 1, 2, and 3, and these are all 10 subunits or more at the time, and far more complex than the bacterial polymerase. So the enzymes themselves said life is going to be more complicated in animal cells than in bacteria. With the advent of the general initiation factors, it's another layer of complexity. But this is all the basic machinery without regulation.
Right. You and many, many other people around the world have, since the, I guess 1980s at least, concentrated on proteins that have some role to play in regulation. And just to be telegraphic about it, there are proteins that largely direct the rate of synthesis, and there are proteins that connect the machinery that the 44 polypeptides you've just talked about, with the regulatory factors. That has been an especially rewarding chapter for you in the last 5 years or so, and perhaps one that you could tell us about because of its great importance and general importance.
Right. So just the
mediator.
Right. A word on the history of that, though. We certainly assumed when we had the polymerase and the general initiation factors in DNA, and we had it working, that we could now look at the issue of regulatory factors. And although we had gotten into that with the 5S gene specific factor TF3A, which was the first of the many hundreds or thousands that are now known, most of the field began characterizing those regulatory factors because they could be done easily by DNA binding assays and various cloning methods. But analyzing those factors in the purified systems, in the case of RNA polymerase II, did not give the elevated or regulated transcription that we saw in crude systems such as nuclear extracts. And that simply said there is another layer of complexity that we certainly had not anticipated. And that came in two forms in terms of the types of factors, but to what, in focusing on what you alluded to with the mediator, in 1991 in animal cells we had seen an activity that was needed for the factors, for the 44 polypeptides in any of several activators to work, and this had also been seen in yeast systems by Roger Kornberg and by Rick Young. We didn't know at the time that our activity would turn out to be the same as the yeast mediator, but this proved to be a complex of 25 or so polypeptides that wasn't needed for a given DNA binding activator to communicate with the basic machinery to make it work more efficiently. Mm-hmm. And as you know, that's turned out to involve interactions of specific DNA binding activators or repressors with specific subunits and interactions of the mediator with the RNA polymerase and the basic machinery. And that, I think, is the major means for communication between activators and the machinery, albeit there are other, sometimes more specialized. So those were cofactors that operate in our purified systems with DNA templates, but the other large group of factors that are equally important are those that modify chromatin, which operate upstream. And although we've been involved with those, they were really discovered by other people.
All right. So we've ended up in this odyssey with 4 groups of proteins, the 44 or so that are general transcription factors, a major coactivator that's 20 proteins, and there are dozens of proteins involved in chromatin remodeling, and then thousands of proteins that are involved in directing the whole show. We can't go on forever, but there are a couple of more things I would like to hear you out on, and I'm sure anybody who is watching this would like to hear it also. You and all of the other colleagues around the world have uncovered what is the most complex machine so far uncovered in all of biology, that machine that makes RNA in a specific way at a specific time. Where do you think the biochemists of the future or the molecular geneticists of the future can look for some relief in this complication so that we can try to tell the world how control is brought about? What can we hope to see in the coming years that will simplify things sufficiently so that we don't have to deal with thousands of proteins to understand things? Right. Is there any hope for that?
Well, I think one of the questions, given this incredible complexity of general factors and cofactors, one of the important questions is whether there's any redundancy in that system. Do we need all of those factors on any given gene? So I think we still have some bookkeeping to do. I think one of the important approaches is now to try to couple our in vitro mechanistic studies with complementary characterizations of genes of interest and importance in the cell, to look in the cell to say what factors are on that gene during an activation process. And this, of course, involves the common chip chromatin immunoprecipitation assay. That, I think, will tell us at least what the likely players are and whether these factors are really there en masse or whether there is some specialization going on. Certainly in terms of the chromatin remodeling factors, I don't think you're going to find all of these there, but it also gives you some idea of the kinetics of their appearance. In terms of one of the things we'd like to do is to, and we've talked about this ourselves between you and I, is look at natural complicated enhanceosomes, that is aggregates of DNA binding regulatory factors that function in a cooperative manner, to really understand the mechanism, because I think we don't know that yet. And the question is, can we reproduce those functions in our test tube assays, even beginning with chromatin templates? So then we have 2 issues there. One is, can we reproduce the phenomenon on a more natural template? And since I mentioned enhanceosomes, it brings up the issue of an enhancer. And of course, many of these are not just a few hundred nucleotides away, but 1,000 or 10,000 or 100,000 in case one of the genes. I don't think we know how distal enhancers work. And I don't know whether we can ever reproduce their function in a test tube, but I would like to try with some moderately far removed enhancers to see if we can really understand the mechanisms, the dynamics of the formation and the function of the pre-initiation complex. So that's part of your question in terms of, I think, what the biochemistry, I still think there's a major challenge on the biochemistry. Of course, I think one of those is the biophysics, the structural analysis. We've proceeded from initial very beautiful studies by our former colleague and my collaborator Stephen Burley on the pre-initiation complex with some of the early events to a point where Roger Kornberg and colleagues and our own Seth Darst in the bacterial system have exquisite pictures of the large polymerase in action on DNA. And the question is, can we extend that to a structure and analysis of the dynamics of the pre-initiation complex?
We've covered a lot of territory, and tomorrow we get the chance to applaud you and to listen to your response in winning the 2003 Lasker Award. Congratulations. It's been a pleasure to be with you this morning. Thanks.
Thank you, Jim.